measure CFOS. It's slightly complicated, but for the sake of this talk, just think about it as a black box mechanism that can measure CFOS, which is, of course, a neuronal activity marker. Whenever you have a higher level of neuronal activity, that leads to higher levels of CFOS, and that allows you to measure whether the neuronal activation has happened. So you find that if we connect the RMAs to the circuit, we're able to have several fold changes upon chemogenetic activation of neurons. And the fold change is actually very similar to what you are observing with the histology. If you were to kill the animal, do the postmortem analysis, you would actually have very similar fold changes as with the blood test. But in this case, you can keep doing that for however long you want, right? So you can actually measure the same animal longitudinally. And this particular CA1 that I was talking about, this is the, now we do have a blood test for the neuronal activity in CA1, as I teased at the beginning. Are there any questions so far? I want to check if we are still on board. No? All right, sounds good. And is everybody hearing me correctly? Can I have thumbs up? All right, sounds good. Just making sure. All right. All right. So let's stop here for a second and talk about what is the future of that, right? So this paper came out a couple of years back. And it's very useful. People are beginning to use it for making, let's say, a blood test for pain levels in a mouse. We're beginning to look at addiction because it's not possible to tell whether a mouse that you killed will have been addicted in the future, right? So people do want to look whether there is a correlation before pre-addiction and post-addiction gene expression. We have made a monkey version of this and lots of different applications, but it is not perfect. And let me tell you about something about scalability of technologies. So I typically ask who here knows about zinc finger nucleases. And whenever you know about them, that means you are slightly older, right? So these were the nucleases that allows you to break the double strands of the DNA, and people are using them for the gene editing. So this is kind of like before CRISPR came to be, that was a hot new technology. But you needed to make a bespoke protein for every sequence that you want to target. And that was not very scalable. In comparison, CRISPR became much hotter, much bigger, because all you need to do is to co-express a little RNA, a guide RNA, and then you can target whatever you want. So it's just much easier to use, right? It's much more scalable. Antibody arrays is a similar situation. You have to have a bespoke antibody for each protein, and people do use them, but not nearly as much as the next-gen sequencing, where you just have to program some primers, and then you can just measure any kind of gene you want. So RMAs are in the way the same, because you need a bespoke promoter for each RMA color. So first of all, you have to have colors, so you have to have different luciferases or fluorescent markers or maybe antibodies that you can use for detection. But you also have to tie the gene expression to a specific promoter. And there's a limited number of those promoters, and there isn't that many of different things that you can track right now with it, right? So again, it requires bespoke promoters and bespoke barcodes, which means that is not very scalable. So what we need is we need to start making the RMA scalable, and we'll start by making programmable gene regulatory elements, right? So we're going to try to make the jump from the antibody arrays to NGS just in the in vivo scale. So this is the paper that just got accepted last weekend in Nature Communications, where we are trying to measure the in vivo transcript levels. So think about it as an in vivo qPCR, right? What we have done with RMAs is more like in vivo Western blot, and now we're going to the scalable, programmable in vivo qPCR, where you just look at the sequence and you can measure a transcript level in vivo. And it uses a recent discovery of the RNase-based sensors, and they are slightly convoluted. So let me walk through it slowly. So you make a single RNA sequence, left hand of which has the sensor that is complementary to mRNA that you want to detect within the cell and has a little stop codon within it. And then downstream from that, you have a P2A sequence that breaks the protein into two, and you have the marker, the output of that sensor. Whenever there's some kind of cell mRNA that is complementary to that sensor, they hybridize together, they make a double-stranded RNA, and there's an enzyme within the cell called ADAR that comes in and edits that stop codon into the tryptophan. When that becomes a tryptophan, this mRNA actually is capable of translating fully. This P2A sequence breaks the translated protein in half, and what we get out is actually just the output, in this case, the RMA or the serum marker, right? So as this progresses, you produce the serial markers, and the more serial markers you produce, the more of these double-stranded RNAs you had, which means there was a higher level of the transcripts, right? So in a way, you measure the levels of the transcripts with the amounts of the markers in the blood, and the levels of the transcripts can be measured by targeting with a defined sequence. So in the same way that you make the primers for the qPCR, you just make a 300 BP sequence for the sensor, and then can actually detect the transcript in cell. So that will we got it too. Sorry, this is from ARIA meeting.org. So this is our UK project that I had the talk yesterday that UCL talked about. It's we're using those sensors in there. So that led to the development of the measurement intact tissues, or MINT, and we find that we can measure the transcripts in the brain no longer with tens of thousands of fold signal, but we still get hundreds to maybe 1,000-fold signal. We have decent selectivity of these, and this is frankly enough. 10,000 or 100,000-fold signal is great, but honestly, for detection, you just need twofold, and anything above it is just a cherry on top. So we're able to actually detect a large array of the various proteins. We have tested ARC and CFOS and VGAT and TH and even some transgenes like TDT. In each case, we're able to detect them in vivo with this kind of sensitivity. We also are able to multiplex it. So now we're beginning to move towards this multiplex recording. Of course, our goal eventually is to have non-invasive transcriptomics. Right now, we just used 3 different kinds of luciferases. We encoded different parts of the brain, delivered those RMAs into different parts of the brain, and we find that we can, with a single blood sample, measure the activity of a network or activity of multiple different regions of the brain by connecting the RMAs to, in this case, ARC sensors, one of those neuronal activity markers. Okay. We also wanted to see if this is something that we could use in large animal. And one of the advantages of RMAs is that they are rational design, right? Which means that you can just redesign them if you want them to do something else. And in this case, what we've done is we swapped the Fc region of the antibody from a mouse version into the monkey version, injected that into the animal, and found we can actually detect the crossing through the BBB, and we can detect the markers in the blood. So in a way, it worked. It was a very interesting story because we couldn't get this funded. We wrote the grant 4 times, and once we got 21st percentile, which was close to getting funded, reviewers had a bunch of comments that we needed to address. We addressed all the comments, and then they ended up triaging it and giving the lower score. So funding that has been difficult, so we only had one shot at making it working, and we basically designed the molecule from scratch on paper, try to make sure that it's going to work according to the way that we understand it, and it actually worked. So we managed to inject the AVs into the monkey brain. We found out that you can monitor the levels of expression of a blood test, which allows us to actually track the gene therapy in the brain using blood test. This is very useful because most of the time you don't have a feedback on the gene therapy expression. So we actually have been contacted by a pharma company that studies the retinal gene expression, where they're trying to use it to check whether the therapy has actually been expressed. Because if the delivery has failed, then their therapy might fail in the clinical trial, not because it doesn't work, but because they cannot tell that the surgery has not been effective in delivering of the genes. We also made an interesting small discovery about AV biology, which is that when you inject AVs into the brain, everybody expects it to slowly accumulate over weeks. And it takes a couple of weeks, maybe 3 weeks, to start seeing the expression. We are seeing that earlier because the readout is quite sensitive. But curiously, we see a spike of expression just 24 hours after delivery. And as far as I know, that has been unknown. We have some theories about why this is happening. We think that it could be that because the AAV is a single-stranded DNA virus, it's possible that if multiple molecules, multiple capsids of virus enter a single cell, they could hybridize and make a double-stranded version that can be expressed, at which point the AAV enzymes come in and make a second strand synthesis, which progresses as the natural thing. But this is important because if you remember from immunology classes, if you have a spike of expression at one point, that creates the antibody response, which then is potentiated whenever there's a second exposure. That's, of course, why we have to have a couple doses of the vaccines, right? So this actually could be quite important for the immune response against gene therapies, most of which are using AAVs. We have found that the BBB in the retina, the BRB, blood-retinal barrier, works very similarly. And in this case, we have been able to detect the gene delivery to the eye. And we also were able to detect the implantation of stem cells. So you can deliver as few as 100 stem cells into the retina, and you still have nice detection. And we do know that some of the stem cells probably die. So it's kind of consistent with that, low tens of the detection limits of the RMA. All right, so let me switch to one more thing. I told you that I would love to have markers where you can know where they come from. And so far, all the RMAs were the method for measuring the invasive gene therapy. You have non-invasive measurement, just like a blood sample, but you do have to deliver them surgically to have the spatial precision. Of course, you could go and measure the entirety of the brain by just delivering an AV permeable AV, right? That big permeable AV that goes into the entire brain, but then you will not get that spatial precision unless you encode it genetically by targeting a promoter for a specific kind of cell type that you're interested in, right? But I did want to have something that allows us for spatial precision. Typically, if you want a spatially precise readout for the gene expression, you would have to do something like a biopsy, which of course break the blood vessels and spills all the blood and cuts the axons and so on. And it's just not ideal because then you just damage that site. You cannot measure it more than once. So that's a problem. So what we did is we found a method how to have spatially precise measurement after delivery of the gene non-invasively through the blood sample, so through blood delivery, to the entire brain. So what we do is we take a BB permeable AV, in that case that was PHPB, we deliver that through the blood, it goes and selectively transduces the brain, and then we simply use focused ultrasound to loosen tight junctions in the BBB for a period of time. That's a technique that has been clinically used. We've worked a lot on it in the other half of our lab, and generally seems to be quite safe and quite effective. And we redesigned our synthetic markers so they can no longer cross through the BBB when it's closed, but can exit whenever the pores happen, right? And the 25 nanometer pores that ultrasound can do between the blood vessels rather than within the cells allows for the exit into blood. So then we simply take the blood sample after delivery, before the opening, then one after the opening, and we see a bump in gene expression that allows us to know that the bump has come from the site that we opened with BBB, right? That we opened with the focused ultrasound. So if we open, let's say, 8% of the brain volume in all the sites that we measure in this case, we have about 30 neurons transduced after that non-invasive delivery. We find that every single animal in the study actually had a higher level of the markers after opening, and that's about a 6-fold change with the arithmetic mean. And that was very significant because every single animal basically reacted in the same way, which I think is very valuable, because if you were doing that in the clinic and you want individual patient to have the result, you don't want to be noisy and better on average. You want every single person to have a measurable effect. We find that the effect is temporary. So if you open the BBB and take the sample of blood before and after, you will see a bump, and then it goes back to the baseline, which means you can look at different kinds of brain regions on different days, which means that you can actually study the entire brain over time and how the gene expression changes. We have found that there's a big dynamic range of the gene expression. Depending on what kind of doses of AV you give, you have different levels of expression, but the fold changes actually stay consistent, are only dependent on the intensity of BBB opening and the volume of the BBB that you're opening. So the signal levels overall can change, but the fold changes of before after opening do not. Those are dependent on the focused ultrasound itself. And then if you wanted to make sure that we can measure endogenous gene expression, exactly the same experiment as in the last case, we connected the synthetic markers to the CFOS circuit, and we found also that we wanted to measure the spatial precision. So we expressed the markers only in one side. So we expressed the markers everywhere, but we expressed the inducers in just one side. And then we activated the CFOS in one part of the brain. And then we found that if you targeted ultrasound to that site that has been opened versus the contralateral one, then you have only the increase in the levels of the marker if you activated the neurons and if you target the site. So it's not like the markers from the opposite sides of the brain are diffusing here. So you do actually have a spatial precision. The fold changes in this technique are smaller than in the histology. As you can see, it's about like 2 or 3-fold lower, but they're still significant. And this is particularly close to my heart because that result has led to our first clinical trial. So we teamed up with some clinicians from the Baylor College of Medicine next door and MD Anderson Cancer Center, which has the equipment for human BBB opening. And a few years ago, we got funding for the clinical trial. It's still being set up. It takes a lot of effort to actually get all the approvals, and find the people and recruit the patients. But it's slowly happening, where we'll be opening the BBB. in the patients, taking their blood sample before and after, and we'll actually be looking at the levels of the endogenous markers that are within the brain in order to understand what changes during progression of PD. When the Parkinson's disease progresses, there's some proteome changes that may be interstitial, that may be recoverable with the focused ultrasound, but nobody knows what they are because they're not going to go into the person and cut a piece of their brain out just to see what kind of proteomics their disease has, right? So if you wanted to study a real human disease as opposed to some kind of animal model where they artificially killed dopaminergic neurons with a drug, then you don't have any other option. So what I'm hoping to do is to measure the transcriptome, sorry, the proteome of the serum after the opening of BBB. And based on that, I want to see what kind of proteins are elevated that are unusual, that are not present in, let's say, less symptomatic half of the PD brain. And based on that, I think we can start making the drugs that are made for humans, as opposed to some kind of artificial animal model where the neurons are being killed with a chemical, right? So based on that, I'm hoping to understand the actual molecular pathogenesis of PD, and based on that, actually make disease-modifying drugs. So that's the goal. We have a recruitment of 10 patients funded, and we'll try to get it going eventually. All right. So where are we going with the whole RMA technology? So in 2022, we developed this RMA's basic technology that can monitor promoter activity. Took us 18 months to publish that because Nature is just very slow, the whole Nature publishing group. Then we made the spatially precise RMAs, and that took us another year or so. And then in the last year, we have published a bunch of preprints, and eventually they got published. Most of them got published in various places, where we developed this non-invasive qPCR, trying to make the measurement of transcript scalable. We have developed fast RMAs, which means you can erase them from the blood to suppress the baseline, and that actually allows you to speed up the time scale of monitoring. We check them in eyes and develop a translational non-human primate version. In the future, want to make massively multiplexed RMAs, so we are developing barcodes that can identify various RMAs and then connect them to various genes using this non-invasive qPCR. So you can measure entire transcripts in the tissue with a simple blood test. So with a single blood test, you should be able to just measure hopefully hundreds, if not thousands, of different genes if we are successful in it. There's a couple papers on massively multiplexed RMAs where we developed 2 different kinds of barcodes that will be coming up this year. Eventually, of course, we want to go from the 12 cells into 1 cell sensitivity, and then maybe one day non-invasive spatially resolved transcriptomics. I'm not willing to put years on that because I think that's still kind of up in the air. All right, so we spent a lot about monitoring. I will have a few slides about control because people are usually interested about it. It all stems from this problem that making therapeutics is very difficult. So, people are scraping the bottom of the barrel for the drug targets. We have apparently reached big pharma. There's nowhere to go but down. We have a broken business model. And worst of all, apparently, is the central nervous system. It takes about 9 years to develop a new drug, and it's only successful 8.2% of the time, despite all the smartest people in pharma trying really hard to make that happen, right? So that's a problem. Drug development is difficult. It goes through this complicated drug discovery pipeline where you start with some kind of understanding of disease biology. You have to get targets in vitro, test them, and then eventually put into a mouse, eventually in a large animal, maybe in human. And part of the problem here is that you make the main decision about the drug discovery in the least relevant system, right? You want it to work in a human brain, but you test it in tissue culture, or you test it in vitro in the solution of PBS. So of course, that cannot translate 100%. That's not a surprise for something as complex as the brain, but this is so far the best we got. So one of the things that we're targeting is to actually redesign this pipeline so it's more in vivo and better, and that's part of my big focus for the next couple of years. But we also are developing this platform therapeutics. The idea being that if you have to go through this process, why not go through it once, develop a therapy that can be applied to multiple kinds of disorders, so you can only go through the last couple steps, only optimize it a little bit, change it slightly, but do not go through the entire thing at the beginning that has a high failure rate, okay? So how do we make therapeutics where a single drug can be used for multiple diseases, right? Because right now we are generally trying to have one drug for one disease, and that's not very efficient. So this is what I'm thinking. Many of the diseases that we do know in the brain actually depend on neuronal activity. The motor symptoms of Parkinson's disease. If you can modulate activity of neurons with electrical signal, that means that you can actually treat those effects, and we know we can do that. Same for epilepsy. you have possibility of shutting down the neurons in epileptic onset zone, and that's going to treat epilepsy symptoms. Depression, similar story. There is a part of the brain SCC that's posited for the electrical stimulation. And if you can do that, if you can modulate neurons and depress the symptoms, that means that you can actually have a single drug that controls neuronal activity, and just then you put it into right places, and you can treat multiple disorders with just a single drug. So really, control of neuronal activity is the key for controlling the psychiatric and neurological disorders. And if you can target at the right place, then you can have non-invasive equivalent of all these DBS scenarios that people have been trying to do. And so this is how the acoustically targeted chemogenetics came to be. The idea is that you can use focused ultrasound to open the BBB locally. That allows you for delivery of gene therapy into the brain. And that specific part of the brain where gene therapy is being expressed, there's expression of a receptor that responds to synthetic drug. That synthetic drug is kind of special because it has no side effects. It essentially just binds to the receptors that have the gene therapy and nowhere else, which means that a simple drug administration after gene therapy has happened allows you to control the neuronal activity in precisely the spot that you target. And if that is the spot that, let's say, controls the feeding, you could control the anorexia. And if that's the spot that controls the seizures, that causes the seizures, you can suppress it and you can control the seizures. If you want to treat Parkinson's disease, you will target basal ganglia. So with a single drug and a single gene therapy, depending on where exactly you point the focused ultrasound, you can just treat dramatically different diseases. This is the receptor that we're using. It's called DREAD, or Designer Receptor Exclusive Activated by Design Drug. It has been around for a while in the neuroscience research, of course. It's been designed so it no longer can bind to any neurotransmitter. Instead, it can bind to designer drugs. There's several of them, but has a signaling that is equivalent to either activatory or inhibitory receptor in the neurons. So by binding this drug, you can either inhibit or activate the neurons, and that allows you to control a variety of behaviors. It's been literally done hundreds of times in animals. We find that this imprinting of the chemogenetics in the brain can be about as effective as the invasive delivery. So focused ultrasound change in the field conditioning in the control of the hippocampus, in activation of hippocampus during memory consolidation, has the same kind of magnitude effect that you would expect if you were to do surgery and injected those dreads directly into the brain. Of course, we had to spend a lot of time on improving technology. We made new kinds of viral vectors that are specifically optimized for focused ultrasound, can have reduced liver expression and increased brain expression comparatively. We find that it's actually safe to open the BBB in the entirety of the mouse brain. And we're kind of surprised by that because we tried to see how much of the brain can we safely open before the mice lose weight or die or have neurological symptoms. And curiously, we just kept going, and at some point we opened 115 sites and it was still okay. We developed new kinds of protocols for targeting cheaply using 3D printed devices. And then we also found actually that immunomodulation can lead to quite substantial changes in the delivery of the AVs to the brain, which was curious, something like 18-fold change in the efficiency with a single change of the IgG antibody producing cells. All right, so this is just a teaser on the control. I'll be talking more about the platform therapeutics in London in person in June 5 to 7. I don't exactly know which date of this. It's going to be at the Vision Weekend of the Foresight Institute. If you want to learn more about this, there is an interview I did with Popular Mechanics in the US, which talks about how we're approaching making the therapeutics faster by not going through the entire pipeline. And I also wrote a perspective on this whole idea of the non-invasive neuroengineering for the Chemical Engineering Progress, which is American Institute of Chemical Engineering trade magazine. All right, so that's all I have. This is our funding and our students and postdocs in the lab. We are in the lake nearby Rice, and we do have UK funding, ARIA, which has been outstanding in funding very innovative stuff, and also a bunch of other stuff from the private foundations and the NIH in the US and so on. So that's all I have. Do we have time for questions, or are we running out of